Hello and welcome back to Window Seat Studies. In today's lecture, we'll be learning about DNA demethylation. In the last class, we learned about DNA methylation. As well as we learned about different members of DNMT family, DNA methyl transferases. Those are the enzymes which transfer the methyl group on the fifth carbon of the cytosine of the CpG dinucleotides. Okay, so in DNA methylation, just a second. In DNA methylation, what happens is that on the fifth carbon of cytosine, there is addition of methyl group and it gets converted to 5-methyl cytosine. Okay, so traditionally researchers thought that this reaction means cytosine to 5-methyl cytosine. This is an irreversible reaction. So DNA methylation was thought to be methylation, DNA methylation, this epigenetic mark was thought to be permanent mark. Okay, why? Because see here the transfer of methyl group on cytosine, this marks the formation of carbon-carbon bond. So breaking of carbon-carbon bond by any enzyme in the biological systems, this is thermodynamically not favorable. So formation of carbon-carbon bond is favorable, but when carbon-carbon bond is established, it is a very stable bond, and that's why breakage of this bond is not thermodynamically favorable. So at very first, when the DNA methylation and all those epigenetic things were very new, and they got to know that DNA methylation leads to gene repression, they thought that this is a permanent mark and this reaction is not reversible. But then here comes the twist. Scientists got to know about 5-hydroxy methyl cytosine. Okay, and after knowing about 5-hydroxymethyl cytosine, they got to know that this provided new insight to the scientists that the methylation DNA methylation mark is not permanent, but rather it is dynamic. So discovery of 5-hydroxymethyl cytosine gave us new insight, and it proved the dynamic nature of DNA methylation. Alongside those discovery of 5-hydroxymethyl cytosine, which we write as 5-HMC, 5-hydroxymethyl cytosine, there was a discovery of a group of enzymes called TIT enzymes. So those TIT enzymes also those were seen to contribute in DNA demethylation. We'll see how in the upcoming lectures. Okay, so those discovery of TIT as well as discovery of 5-hydroxymethyl cytosine, this led to the very new domain that DNA methylation can be erased.